over to you.
Thank you. It's delightful to see everyone, to be back with the GeomPack folks, and this is a really fun topic, and I hope it's of interest to other people here. It's a biological system that I came across at a very fortuitous time, so I was not really aware of the fun of packing until I took my sabbatical at the Newton Institute, and there was a magnificent talk by Dennis Weir introducing me to the fun of packing problems. And right after my sabbatical, I came back and was approached by someone from Duke just down the road. We're very close. The rate limiting step on collaborating with people at Duke and UNC is simply finding parking. It's otherwise very doable. And Michel had this system he was working on. He's a biologist, the development of the notochord in zebrafish. So zebrafish is one of the sort of white mice of developmental biology. We like to use it because the embryos are transparent, so you can see an awful lot that's going on. And you hear fish in some kind of deep evolutionary sense. And the notochord is a structure, it's not just in fish, it's in any creature with a vertebra, that is to say all of us vertebrates. It's also there in non-vertebrate chordates. You may not think about us having cousins that are invertebrates, but if you go to the shore, and reach down under the dock, you may find squishy things called sea squirts, which are fun for your kids to squeeze and squirt each other with. And they are also chordates because when they're young, they have these notochords, which start out very small and they elongate, like blowing up a long balloon for folding into a toy dog or something. And what they serve to do is to take a very round embryo and elongate it. And as the organism is maturing, the spine forms around the template of the notochord structure, which has made the embryo elongated. This was a fun topic for the dissertations of not just that first student, but a second student, and they have both long since graduated, and Emma's working in industry and Evan is postdocing in New York. And let me tell you the rest of the story, because it's about the packing of the cells inside the notochord at a particular stage of development in the model system of the zebrafish, which is the one we know the most about. There are studies of other fish embryos, and there's a small amount of comparison to be made, but let me give you a little bit of an anatomy lesson. At this developmental stage, the notochord is made up of rather large internal cells with large vacuoles. Almost the entire cell is made up of this bag of gel, essentially. And so mechanically, we're going to think of it mostly as a bag of gel. The internal cells called osteocytes. I always get them backwards. The osteocytes internally, they're held in by a sheath, which has cells in it, which are very flat. They're squamous. Those are called osteoblasts. And mechanically, that sheath is dominated not so much by the properties of the cells, but mostly by the properties of the extracellular matrix that they're attached to. We're mostly, for these purposes, going to ignore the mechanics of the cells in the sheath and just think of the sheath as a mechanical continuum, and focus on how that interacts with the packing of the cells inside and the mechanical interactions between the sheath and the internal cells. So I'm no longer going to talk very much about the cells in the sheath, the osteoblasts. And the cell packing is very stereotyped at this stage of development. We tend to see the cells packed in a zigzag pattern, which we're going to mostly call staircase. You'll see a few exceptions. And if you look at the microscopy, of course, the cells are not uniform in size. And this can cause some analytical challenges when we're looking at the data. But we can certainly relate the cell packing to some experimental observations in nearby systems and some analytical work that we can do. Why it's important biologically to the development of the notochord is kind of interesting. So if the packing of the cells is disrupted, either because you have a mutant or you go in and zap with a laser some of the internal cells, this is very distressing to the structure. And these sheath cells, which I said I was mostly going to ignore, they will migrate in to fill any gaps and differentiate to form more internal cells to seal it up and have the correct number of internal cells. So it's a wound healing response designed, it seems, to restore the cell packing in the interior. If the normal structure of the notochord is messed up by some of these interventions or mutations, all kinds of bad things happen, like the buckling of this tube. You have trouble elongating, you may get a short embryo, you may get scoliosis in the spine because the notochord has not maintained its straightness and has buckled. And so it seems to be that it's biologically fairly critical to maintain the integrity of the notochord at this stage, and as the mechanism for doing that, maintaining the cell packing and the pressure. We good so far? This is of course the GeoMPAC group, so we're used to thinking about packing, but I am agnostic about the mechanics of these notochords, and so we were looking. Why am I agnostic? Because it's almost impossible to measure the mechanical properties of these tiny things, because as soon as you go in and measure, you've destroyed the thing. So we have thrown several different mechanical models at trying to understand what's going on in the notochords, ranging from a hard sphere model to a soap film model to elastic membranes, where I'm going to mostly talk about a soap film metaphor and an elastic membrane metaphor, but we were also looking at soft spheres, which you're going to see fairly soon in the experimental portion. There's, of course, a huge body of literature on the crystallography of packing regular patterns in cylinders, and there is the phylotactic nomenclature where you have an integer triple referring to the neighbors in neighbors of each unit in 3 different directions. I'm mostly going to stick to verbal descriptions and call this lowest order pattern bamboo, and the second lowest order pattern staircase. The other low dimensional patterns, the next one we might call the chiral pattern. We're mostly going to be looking at those 2 lowest order patterns. In the bubble context, the foam literature, which pattern you see experimentally is governed primarily by the length ratio, number of units, bubbles, cells, what have you, per unit length, and where that is also equivalent to a ratio of diameter of the tube to diameter of the sphere equivalent soap bubble. And there's all kinds of work which people in this group have done, and their trainers and so on, establishing in simulations and experimental systems where the patterns are observed. And the thing to note here is that there is overlap in the range, that there can be at particular lambda ranges, you can observe more than one category of pattern. And we're going to be seeing this overlap region manifest in coexistence of the patterns even within a single tube of cells. Because the natural patterns in the notochord are not regular. They are lab experiments, but they're not controlled lab experiments. We're not imposing a particular regularity on the patterns. The cells are different sizes from each other. They may have different amounts of gel inside each cell. There can be irregularities in the sheath. If you look closely at this microscopy, you'll see that the whole structure itself is not of uniform diameter. It's tapered. The ends are narrower than the middles. And we looked at an analogous system for assistance where you can buy these gel beads for flower arrangements, and they have all kinds of different brand names. These were jelly beads, and you can stuff them in a tube. These happen to be in a plastic bag because this was much more accessible to me. And depending on how many you pack and how much you hydrate them, you can study the patterns quite cheaply and conveniently, and have, depending on the diameter of your tube and the diameter of your gel bead, different patterns in your office. And so we studied in a more controlled situation than my office, the lab, that is to say. We were packing known numbers of gel beads in a tube to maximum hydration, and analyzing the patterns in the tubes of known diameter, characterizing them for all kinds of measurements, in particular the geometric ratio, and comparing with the equivalent analysis in the notochord systems. But again, this is a statistical analysis rather than looking at regular patterns, because there's coexistence. Well, in order to do that, we had to first define, well, what is meant by a pattern that's not completely regular? Because there are transitions in the patterns. I mean, you can call them defects, but if they're happening one after the other, bamboo staircase, bamboo staircase, bamboo staircase, is that its own pattern? So we had some patterns which seemed to be repeating quite a bit, like 2 and 1 and 2 and 1, which are not something you would see in the soap films in rigid tubes, but we were seeing them in the gel beads, and we were seeing them in the notochords. We called that one hopscotch. So we had to come up with criteria for, is this transition, what is this transition between the particular patterns that we're seeing? And going from the microscopy to the analysis involved a lot of steps. So the segmentation is fairly standard. It's not trivial. It always requires some human intervention, because you get these little fragments. Are they cells? Are they not? You have to make a decision on them. So the segmentation, there are macros already for finding the centroids and the volumes. That's fairly easy. But then we needed to mathematically straighten and rescale these things so that we could do a comparison along the length within a single notochord, which is tapering and so on, and where the cells may not be of equal volumes. And that allowed us to calculate the lambda as a function of position, characterize the local patterns, and so on, and then be able to do the statistics on the patterns that we were observing. And the gel bead systems were much easier because we didn't need to do any microscopy. You could just hold up the tube and count. And the diameter was consistent with the gel bead tubes. So for the notochords in the lab, the patterns that were observed locally in the different individual organisms along their lengths corresponded very clearly in their distribution to what is seen for what is known from the soap bubble systems and the numerical simulations, corresponded very nicely to the expected values of lambda that were imposed experimentally. And so this was some kind of validation of our hypothesis that essentially the notochord is packing of soap bubbles, or packing of the less analyzed system, the gel beads. And when we compared the distribution of patterns, so we can't control the lambda of the notochord system because it's an animal, but we can certainly control the lambda in the gel bead system by putting in more or fewer beads. But for the average lambda of a particular notochord, we could look at the equivalent gel bead lambda and look at the statistical distribution of the patterns. And it was more or less an equivalent distribution. There's a couple of patterns which were apparently significantly different, but the gel bead system and the notochord system have mechanical differences. The gel bead sheath is quite rigid, and the sheath of the notochord we don't think is quite rigid. So we had to also look at other examples in the and I will get to that in a minute. In the zebrafish, we were able to get a little bit of variation in the lambda by looking at mutants. So the wild type, the normal zebrafish, is primarily packed in the staircase pattern, and all of these blue spheres here are representing locations where we've got the staircase pattern, and the reds on the end are packed in bamboo, and the yellows interspersed here are short segments of a chiral pattern. In one mutant which has fewer cells, it is almost exclusively packed in the bamboo pattern. And in another mutant that we looked at, which has more cells per unit length, we were seeing the bamboo pattern only at the tip, and primarily the chiral pattern interspersed with short segments of staircase. And you could see it along the length. You could also see it in the end-on view that the wild type primarily has its centroids of the cells distributed left-right. I didn't actually hold this up. So in the wild type, not only is it packed mostly in staircase, but specifically the packing is left-right rather than front-back. And that's going to become important later, much later in this talk. But again, for these three genetically different situations, the patterns seen were corresponding primarily to the regions of lambda space that were known to be dominated by particular low-dimensional patterns. There was something that we noticed one day staring at some of the microscopy. I looked at this and I kind of squinted and I said, yeah, is that a trick of the perspective? Is that not exactly a right angle cross section, or is that thing off around? And so we went in and measured more carefully, and it turns out that there is some off roundness to these notochords. I just wanted to get it up here that we are looking at flattened notochords in terms of both aspect ratio and eccentricity. They're both measuring the same thing. It's just one is scaled to 0 and one is scaled to 1. And they are nonlinearly related to each other, but we're going to be discussing here and there aspect ratio or eccentricity interchangeably. They're just zeroed out. They're based on different measures of a circle, either aspect ratio 1 or eccentricity 0. So it turns out that the wild type notochords, actually all of the notochords, are a bit off of round. And in the wild type, which is dominated by the staircase pattern, we have an aspect ratio of about 1.1, and a sizable eccentricity of 0.4, and it's wider left-right and narrower dorsoventral. So that suggests that the sheath is flexible rather than rigid. And that got us thinking maybe we should be looking at flexible tubes in the lab, or at rigid tubes which are oval to begin with, because there were some that were rolled so tightly on the spool that they had permanently gotten oval. So we packed the beads in those tubes. And the staircase pattern was seen over a much wider range of lambda values in the elliptical tubes. So it would appear that there's a bidirectional relationship between the eccentricity of the tube and the appearance of the staircase pattern. Furthermore, we went in looking at flexible tubes, silicone tubes, where we were not predetermining the aspect ratio of the tube, but when it was stuffed with an appropriate amount of beads, it was more likely to be in the staircase pattern. And when it was in the staircase pattern, it was more ovaled. The aspect ratio of the staircase pattern in the flexible tubes was off of 1. So if you have a flattened tube, you are more likely to get staircase. If you have a staircase pattern in a flexible tube, you are more likely to flatten that tube. And so it's going both ways. Where does this come in the notochord? We then looked at different models of the mechanics of the sheath in the notochord to see what is this relationship between the aspect ratio and the eccentricity. Excuse me, the aspect ratio and the packing pattern. And this was Emma Sorrell's dissertation work. And she looked at 3 models. I'm going to primarily talk about the model where the sheath is flexible. But she also looked at a rigid sheath equivalent to the rigid tubes in the lab and a compromise model where it was rigid in the longitudinal direction and flexible but constrained to an ellipse in the cross section. All of these are assuming that the notochord is infinitely long, that we have a fixed number of cells per unit length, that the sheath is uniform, the cells are equal, and the cells are mechanically modeled as equivalent to soap bubbles at dry foam under surface tension, that the patterns are regular. We're going to measure things with cells per unit length, which is imposed, fixed eccentricity, or an eccentricity as a consequence of the packing pattern in these latter 2 models. For the rigid sheath, we don't need to keep track of a surface tension ratio, but for these other 2 models, we were keeping track of the ratio of surface tensions in the sheath versus the interior cells. In the rigid sheath, the eccentricity was imposed corresponding to the gel beads packed in a flexible silicone tube. And in the flexible sheath model, we calculate the eccentricity of the arrangement after the fact. Let me just show you some sample simulations. It's an infinitely long notochord, but this is a finite segment of an infinite simulation. So to do the infinite simulation, these are done in Surface Evolver. We simply assume a period 2 configuration and started with the correct topology and let it evolve to the pattern that minimizes the surface energy. And at a particular lambda value, we get stability of the patterns or instability of the patterns. But I'm less concerned with the rigid tubes than because that's really a cartoon and not corresponding to the notochords at all. With the fully flexible tubes, where the sheath is essentially like spandex hugging each of the interior cells, but it's got its own surface tension independent of the surface tension of the interior cells. As we varied lambda and as we varied the tension ratio, we were seeing different shapes in side view. Not surprisingly, if your sheath is very soft compared to the interior cells, you would expect to see strong cusps where the cells meet each other. And as the sheath gets to be much greater tension relative to the surface tension of the cells inside, the sheath becomes much straighter because the cells inside are accommodating the pressures generated by the sheath outside. the 3D perspective is lovely to look at, but we were then able to quantify the eccentricity as a function of the tension ratio. Of course, the bamboo patterns are not eccentric, they're round in cross-section, but in the staircase pattern, not surprisingly, the eccentricity is going down as the tension ratio is going up, and you get your greatest eccentricity at the lowest levels of sheath surface tension. And then Emma was able to find where the bifurcations were. She was unable to get stability of staircase patterns below a lambda of 1.1 something. There was a third pattern that came out that was period 2. This was really fun. It wasn't quite, it was topologically equivalent to bamboo, but it was geometrically more similar to the staircase pattern. We nicknamed it a serpentine pattern. It essentially is a buckled bamboo pattern, buckled to the left and to the right. You see it in the hard sphere packings. It's really just an artifact of imposing the lambda, because we have an infinitely long notochord, and we have to declare how many cells we have per unit length because there's no ends. So this isn't a real thing in the notochord system. It's just a curiosity that happens to be there, and it's very nice. So in sum, the bamboo patterns are always round in cross-section, no matter what the surface tension ratio. The staircase patterns are always eccentric in cross-section, with the eccentricity going down as the tension ratio goes up. But the problem here is we're using the infinitely long notochord model and imposing the lambda. What's determining the lambda? So it would be nice to have a model with fewer parameters. I mean, I can work with 3 parameters. That's okay, but I prefer a model with 2 or even 1 parameter. So we looked then at a model that doesn't predetermine lambda, because we're using a finite length notochord with a finite number of cells. And we looked at a broad range of n for the cell packing, and we were not assuming periodicity of the minimal energy solutions, but finding minimal energy solutions in a particular topology for a particular tension ratio and a particular n number of cells. And I just, this is one of my favorite figures, and it's so nice that my student gave me a graduation present when he marched, and this is just the best mug ever. And not surprisingly, when you have very low sheath surface tension, the cells remain almost spherical, and there is a lower limit. At some point, you will break the topology of these setups. If the gamma is low enough, it will break, and so we did not go. We didn't go below that. Similarly, if you have the sheath surface tension overly large, it gets ridiculous because you can never elongate the thing. It's just all compressed into a sphere. But in the intermediate range of tension ratios, it's kind of looking like the notochords. In the intermediate range, we also call this the baguette display. It's like a good bakery. So once we had this range of minimal energy configurations, we could do a little bit of analysis on that. And as the pressure varied along the length, that was allowing the ends, because of course this is a soap bubble model, and the pressure relates to the curvature. So the ends are going to have a different pressure from the cells along the length, which have a more uniform curvature equal to each other, that is. And the pressure was uniform no matter how long the notochord was, until a kind of end zone. And we were able to also quantify, well, how long is that end zone? And that's a function of the tension ratio. Fair enough, nice enough. Likewise, we were able to confirm that the aspect ratio of these structures in bamboo was always 1, that the aspect ratio of the staircase configurations was always greater than 1, and very uniform in the interior, slightly tweaked towards the ends, as you might expect, because the curvature is changing. The two curvatures are changing. We were able to quantify the aspect ratio as gamma varied, as we know it's getting closer to round as we have a higher sheath tension. And interestingly, this curve passing through the data was a really simple expression from a simple 2D model of just two circular arcs and a straight arc between them. So that wasn't a curve of fitting. That was a curve of a simple formula from a different dimension of model. And similarly, a real cartoon of how spheres pack in zigzag. If you have rather than a staircase, a plane between them, gave us some really useful formulas, and the data passed along the curves of those formulas derived from a simplification of the geometry of the zigzag configuration. It was very lovely to tell us what the lambda was going to be as a function of the tension ratio, and what the pressure was going to be as a function of the tension ratio. It was a very clean result and quite satisfying. So we could get the same results without having to impose the lambda, but it's still not quite answering any biological questions yet. It's answering some packing questions, but it's not really answering the question of, why do we need this notochord to be intact? Why do we need the ellipse? Why do we need the eccentric cross-section? Why do we need the packing pattern to be staircase? And we have to return to the function of the notochord, which is to be rigid and elongate the embryo and stay straight while the spine is being formed around it, while the spine is being built around it, and not buckle like this obnoxious advertisement that I don't know about. Do they have those in the UK? They're pretty awful here at every car dealer.
We have them.
I'm so sorry. So to examine the mechanics of the notochord, which are fairly difficult to analyze experimentally. It's very hard to measure these things, because if you take them out of the fish, they're not intact. You can bend a fish, but you're bending all kinds of tissues, and not just the notochord. So the model is going to help us understand why it's mechanically important to maintain the observed structure. And so we took not the minimal energy surface geometries that were generated by surface evolver, but rather we used the geometric ratios from the analysis to construct a CAD model in order to do a bending study. So these are not minimal energy structures, but they're really quite close, because we were able to get such satisfying agreement between the cartoon geometry and the minimal energy geometry that gave us confidence that we could use just the ratios in the cartoon geometry to construct a CAD geometry. And then vary the internal pressure, vary the cell count, the packing pattern, the ratio of, well, not surface tensions, because if we're going to bend the thing, imagine you have two soap bubbles that are touching and you bend them, they're just going to roll on each other. That's not a well-determined system. So we had to switch from a soap bubble model to an elastic membrane model where there is a reference configuration. So we were no longer looking at ratio of surface tension, but looking at ratio of elastic membrane tension. Okay, to have a reference configuration that we could bend. So before you can bend it, you have to inflate it, because to sleep on your air mattress, You have to fill it full of air in the first place, and you have to have a reference configuration before you bend it. So we first had to do an inflation study and varying the pressure and the stiffness ratio, and finding that the inflation was isotropic. There, because we had not imposed any anisotropy in the material, there was no anisotropy in the inflation. It retained the same geometric ratios. The strains were isotropic. And we got a really kind of complicated relationship between the internal pressure and the stiffness, the membrane stiffness ratio. And we struggled for a very long time with how to display it, but suffice it to say that as the sheath gets stiffer, the overall bulk modulus goes up, and there's a secondary effect from the excuse me, I said it backwards. As the pressure goes up, it gets stiffer, not surprising there, which is what our experience from the air mattresses, and it has a secondary effect from the stiffness ratio. Once we understood how stiff it was in inflation, we could then do a 3-point bending study to determine its flexural rigidity. And the displacements we found were very much in agreement with classical beam theory. And so that allowed us to simply look at the maximal displacement as a function of the membrane stiffness, the internal pressure, and the packing pattern. And with that, in order to understand whether there was some advantage or disadvantage to the different packing patterns, we needed to do a fair comparison. So if you think about it, you've got 20 cells, you pack them in bamboo under a particular internal pressure, there's a particular stiffness you should expect. If you take the same number of cells and pack them in the staircase pattern, think about it, it's going to be shorter and fatter. It's going to have a much greater flexural rigidity. A short, fat stick is harder to bend than a long, thin stick, even of the same mass and same material. So we had to make a fair comparison. And of course, there's engineering formulas for the moment of inertia of tubes of various cross sections. And we looked at a thin-walled elliptic tube, and there are exact formulas, and that's going to depend on, of course, the thickness of the tube, but also the aspect ratio. So a round tube has a reference moment of inertia, and as we go off of round into the eccentric oval tubes, we're going to have different moments of inertia in lateral bending and in dorsoventral bending. So flexural bending and lateral bending will have different moments of inertia, different flexural rigidities for the comparison system of just a hollow cylindrical oval tube. And as you increase the aspect ratio, there's going to be a greater and greater difference in the stiffness in lateral bending versus flexural bending. Naturally, the staircase is going to be stiffer than the bamboo for the equivalent number of cells and the equivalent mechanical inputs of the stiffness ratio and the pressure. But as we get the greater and greater eccentricity, the greater aspect ratios, we see a greater difference in the stiffness in the two directions. Okay, so this is our reference curves here. And when we look at the simulations, the reference curves are quite parallel to the data from these 3-point bending simulations, although they underpredict how advantageous it is to be packed in the staircase pattern. We actually found that the notochords packed in the staircase pattern were not just stiffer than the equivalent in number of cells packed in bamboo, not just stiffer than those, but stiffer than predicted by the hollow tube, which was quite interesting. So what's it coming down to? If you've got a notochord and you need it to be stiff, it's going to be stiffer when it's in the staircase pattern. And it's going to be much stiffer in the bending direction this way. Because remember, these notochords are in the zebrafish with the zigzags oriented this way. Okay, it's longer this way, which means it's stiffer this way. It's softer this way. Think what happens if you're trying to elongate the embryo and it buckles, and you're going to wind up being a fish that's kind of hunched over this way. Okay, that's not great. If you buckle in this direction, you're going to be a fish that's permanently bent in this direction, and you'll be swimming in circles. You will not get very far in life. So there's an obvious advantage to being stiffer in this direction than in this direction. You elongate straight. What's cool to me is that the staircase pattern is the only one of the patterns. It's the only one of the low dimensional patterns, or even the high dimensional patterns, in a flexible sheath. Everything but the staircase pattern is essentially round. Everything is going to have the same stiffness in all directions. This is the only one with differential stiffness, depending on the direction of the bending. I mean, it's a simple mechanical consequence of packing equal items in a flexible sheath, but it's there as this thing that genetically is being clung to in these fish. They have all kinds of mechanisms for fixing it if it breaks. So, I mean, maybe this is a one-off evolutionary development. I think it's very lovely, and it's been really fun playing with this. And I am eager to hear people's questions.
Oh, that's really fantastic, Sharon. Thank you so much. I'm just going to stop the recording and invite people to ask